Good morning, viewer. Welcome to our Glory Globe TBS breaking news. Yeah, transposons DNA sequences that can self-replicate and move, that means jump throughout the genome, are widespread and can affect cell survival if left unchecked. So cells control these jumping genes by silencing them, but little was known about how cells recognize and defend themselves from invading transposons. St. Jude Children's Research Hospital scientists have answered that question, showing that cells sense abnormal RNA patterns produced by invading transposons or respond by activating pathways to silence them. Further, this process extends to any invasive DNA, provided it produces enough RNA disturbance for cells to detect. The study published in Nature Communications offers insight into a genetic phenomenon that is at the center of diversity and evolution. Led by corresponding author Mario Halik, PhD, St. Jude Department of Structural Biology, the researchers found that fission yeast cells silence invading transpositions using two pathways. The first is RNA interference, which silences genes by destroying their messenger RNA. The second mechanism of silencing utilizes heterochromatin, a highly condensed form of DNA, which physically blocks transcription factors from engaging the DNA and therefore halts gene expression. Beaver cells use from RNA interference and heterochromatin to detect and silence transpositions with recognition efficiency based on insertion location where in the genome and transpositions is and copy number, how many copies of the transposition are there. While the work was conducted in yeast, similar defensive mechanisms are also likely to exist in higher organisms, particularly in germline cells, that means sperm and eggs, which are particularly vulnerable to transposition induced in disruption. Every organism tries to defend itself from transposition invasion. They can proliferate uncontrollably and occupy large parts of the genome, slowing growth and negatively affecting gene expression. Halik said, defensive systems like these are typically restricted to germline cells, where strong defense is essential. Otherwise, which transpositions could propagate dramatically within just a few generations. Beyond this exclusive report, Glory Globe to Delhi, Bharat.